Good afternoon, students. Well, actually, it's about 6:30, so it's in the early evening. I'm glad to have you back with me. Uh, we are going to go over lecture 4 in your notes today. I did give you a little forewarning. I wrote yesterday an email so that you'd be prepared for today. I'm going to try and work through a lot of the material in lecture 4 and 5. We'll see how far we can get. But best thing, remember, if you have any questions, to please call me. I will write you another email after this is finished, and I'll list all the pages that I've covered. I did have a couple of videos that I wanted you to watch. They're excellent, and I think about the physiology of the cerebral cortex. And if you remember correctly, and let me move this down a little bit, if it'll go down. In studying the cortex, or the cortices of the brain, it's the outer 1/4 of an inch of all the gray matter of the cerebrum. It's only about 1/4 of an inch, but it's very powerful. We learned the other day that it's divided into a frontal lobe, parietal, occipital, and temporal. So those 4 lobes have those 4 plates of the cranium covering them and protecting them, as well as the cerebral spinal fluid, is responsible for interpreting sensory impulses. That means everything you see, hear, taste, touch, smell, and feel. It's the cerebrum's responsibility to analyze that, interpret that, make sense of that. You have sensory neurons that are carrying the messages of sound and sight and smell and taste and the proprioceptive senses. You have all of this filtering into that brain, and the brain has lots of associating neurons, connective neurons or interneurons, which will link all of this sense and make sense of it for you. So the cerebrum is responsible for interpreting all these impulses, making sense of them, initiating voluntary muscular movements. Everything you do with your skeletal system that's voluntary is going to come to you from the brain. And you've got some beautiful diagrams. One of the things I want to mention to you is a little bit about the brain landmarks. Again, remember the other day we talked about the central sulcus that's on the left and right brain at a right angle, lateral sulcus, the transverse fissure, and we do know that both hemispheres of the brain are divided into the left and the right hemisphere. This area, the central sulcus, has a hillock, a gyrus right in front of it, an upheaval of that cortices. And that gyrus right in front of it is the central sulcus, precentral gyrus. And it's called pre because behind that central sulcus, that landmark, is another gyrus, and it's called the postcentral gyrus. Post coming after the central sulcus. It's a gyrus, so it's a post-central gyrus. And the gyrus right in front is before the central sulcus, so it is the precentral gyrus. These 2 locations are extremely important because the precentral gyrus, this piece of tissue that goes all the way down the flank of the frontal lobe on both sides, is responsible for your ability to control gross and fine motor movements. All of that comes from this tissue. The tissue right behind the central tell me how your shoes and socks feel on your feet, your slacks or tights on your leg, your shirt, how it feels on your torso. That is all a tactile sense. These sensory afferent neurons that are picking up the way these clothes feel on your body, they're sending it right to the postcentral gyrus so that you have cognition, awareness of that tactile sense. So all of this on both sides, the postcentral gyrus, gives you that ability. I can touch the top of my desk. It's solid, smooth, and it's cool to the touch. Sensory afferent neurons are sending the message afferently to the spinal cord. It goes up the brain stem and is directed, we'll find out later, by the thalamus to the post central gyrus. So in this area, you feel, you have tactile sensations. Before that, the precentral gross and fine motor movements. So this gross and fine motor movement, you are sending the messages down the brain stem and down the spinal cord to your muscles and glands. So you have a volition or control of that. Now, that is motor, whereas the postcentral gyrus and that tactile feeling, that is, and it has a lot to do with the entire cerebral cortex working together. But that is memory and reasoning, your personality, really who you are, is right here in this frontal lobe, before the central sulcus, but in this area right in here. And intelligence and personality, which is number 4, comes from this area. So you interpret with your entire brain what you see, hear, taste, touch, and feel. You control motor movements with the precentral gyrus right in front of the central sulcus, and you have tactile sensations from the post-central gyrus. Regions, and the next section is regions of the cerebral cortex, perform specific functions. The cerebral cortex, or again cortices, can be divided into motor areas. Motor area is in the parietal lobe, postcentral gyrus, and tactile senses are in the parietal lobe, postcentral gyrus. Tactile, whereas motor is in front of the central sulcus. I just disputed myself, excuse me. And throughout the entire brain, there are associating areas, linking sensory and motor and making sense of everything that you're experiencing. The motor areas. Motor activity is found in the frontal lobe and the front of the central sulcus. We talked about that. But you also have in the frontal lobe, the lateral frontal lobe, right in this area, you also have, and these are pointed out in your textbook, so please look at that. There's an area in here that its ability was discovered by a Dr. Broca in England in the late 1800s, and it's called Broca's area. It's about the size of a quarter to a 50-cent piece, and this area right in here is responsible for the controlling of your tongue and your lips, all of this movement in your mouth to form speech. You're able to carry that out from Broca's area. That's a motor area. If you, and you will see patients that may have had a stroke or a cerebral vascular accident, and blood was released into this area. Remember, red blood cells never want to come in direct contact with neuro tissue because it's destructive of nervous tissue. Everything is exchanged between the cell membrane of blood capillaries. So if someone has a stroke or a aneurysm or a break of these blood vessels and they ooze out, they can destroy Broca's area. And your patient may know what they want to say, and they're trying to say what they're thinking. They're thinking clearly, but they can't pronounce the words. It'll come out gibberish if this has been injured. Not only can an aneurysm, if it breaks, can destroy that tissue, but if you have a blood vessel that swells and puts pressure on this delicate nervous tissue, it can also destroy it, and that's Broca's area. Most often, you'll see that in the left hemisphere, but sometimes everybody's a little bit unique, and sometimes the Broca's area will appear in the right hemisphere. So all of us are unique in our appearance, and our brain is too. We have structures that are basically all the same, but we do have some differences. Why? Was a science in the 1800s that said you could feel someone's skull, and if you could feel the bumps and maybe the ridges or little depressions in that skull, then you can interpret their personality. Well, they did actually believe that, and they made models, but it has no correlation whatsoever. But that was a concept that has gone by the wayside. That was called phrenology. Okay. Now, if you look at sensory area, below the motor areas, sensory areas occur in several lobes. They are parietal, temporal, and occipital. So the entire brain is involved with sensory. Motor also, but also it's above the lateral sulcus right here. And this is the parietal lobe that has been done in kind of a dark purple. It's above the occipital lobe, and it is, of course, the postcentral gyrus along the central sulcus that the area is involved in tactile sensations, skin of the skin, such as temperature, touch, pressure, and pain. Somatic sensory area in this area. I'm reiterating this. I'm going over this again because it is important. In the occipital lobe, that's this darker blue, almost kind of a turquoise blue right here. The occipital lobe is responsible for the reception of vision. You can have perfectly good sight, 20/20. Your eyes can be healthy, your optic nerves can be healthy, and they're going to this region to deliver what you see to this region, so you have awareness of it. But if tissue is destroyed in the occipital lobe, then vision is destroyed. Your eyes and nerves can be perfectly healthy, but that part of the brain to enter this occipital lobe can completely destroy the vision. But that is the visual receptive area. The next, and in front of the occipital lobe, this area is primarily useful in sound perception. The sensations are not necessarily motor or sensory in function, but they're interconnected. They're interconnected areas, and they are in the anterior portion of the frontal lobe. They're widespread in the temporal, parietal, and occipital lobe. They help the individual to analyze, interpret sensory experiences that are involved with memory, reasoning, verbalizing, judgment, and emotional feelings. These are the things, but you actually think with processes from the entire cerebrum, specifically the cortices. And that is discussed on the next page, on page 14. The thinking process involves lobes of the entire cortex. The right cerebral hemisphere receives information and directs activity on the left side of the body. You have in your right hemisphere here the nerves that pass down through the brain stem. They go to the medulla oblongata, and on the surface of the medulla oblongata, that would be right in this area. The nerves decussate, Decussate. That is on the exam, decussate. And they cross over. The nerves that cross over on the anterior portion of the medulla affect the left side of the body and control motor volition on the left side, and the nerves that cross over on the anterior medulla decussate will affect the right side of the body. So the right side of the hemisphere will control the left side of the body. The left side of the hemisphere will control the right. Corpus callosum. Okay, I mentioned a little bit about this, and I'm going to talk about it again. So I want to get, okay. There's a mid-sagittal section through a human brain. I think I showed this to you the other day. And it's truly a beautiful specimen and for us to study. Here is the corpus callosum. Remember, the corpus callosum holds the two hemispheres together. It's heavily myelinated. That means the oligodendrocytes and the three other neuroglia in the brain are myelinated, and it gives that corpus callosum its white appearance. In order to separate the left and right hemispheres, you have to take your scalpel and actually cut through the corpus callosum, down through the diencephalon, and down through the brainstem. If we were in lab and you were working with your sheep brains, we would be able to do this. But we're going to have to imagine it, that we're actually dissecting the brain. And then, too, we've got some brain dissection videos that are excellent. And we can actually see some human brains. One of the brain specimens is brand new. The patient just died, and they harvested that brain from the patient, and you can actually see a brain as it was taken from the cranial vault. But that is the corpus callosum, and it is above the diencephalon. The diencephalon, remember, is the thalamus, which is an egg-shaped gray stripe, but that's the diencephalon, and this is the midbrain, pons, and medulla. As I mentioned the other day, some scientists consider the diencephalon as part of the brainstem. Others refer to the diencephalon as the tween brain, and they don't consider it part of the brain stem. And I like to mention that to both of you, to both of those points of view. Look right under where it says basal ganglia. I want to explain something to you. And you'll have to bear with me because I do have a picture of it in your textbook. Anytime you see the word ganglia, what it means is it is a cluster of cell bodies. It's a cluster of cell bodies and dendrites that actually make what they call a ganglia. Now, usually, if you talk about a cluster of cell bodies in the peripheral nervous system, they simply dub it ganglia. If you're specific and you're talking about a cluster of cell bodies in the brain, it's referred to as a nuclei. And it is called the basal nuclei if you're going to be super correct. But anatomists, textbooks will use those terms. Basal ganglia exist in the PNS if you're going to split hairs, and nuclei exist in the nuclei exist in the brain or the CNS. Now, the basal nuclei, and you can see them deep within the brain, and there are 4 darkened areas. And these darkened areas, as I said, are just clusters of cell body. To be specific, we call them basal nuclei. And what they do is they release an extremely important neurotransmitter known as dopamine. And dopamine area in front of the central sulcus, precentral gyrus. So they make sure that only the correct amount of acetylcholine is released into the system so that you have smooth motor movements. If something interferes with the production of this dopamine, and if it does, And once it's identified, it's known as Parkinson's disease. That's what um, Michael Fox, I believe that was his name. I can't think of the actor's name right now. And Muhammad Ali, through football, contributes to the development. It can develop as young as the mid 30s, as it did in Fox, and later on in the 40s when it developed in Muhammad Ali. And it was literally in Muhammad Ali caused by punches to the head and concussions. But it releases dopamine, and it's right in this area. Dopamine regulates acetylcholine. Now, if it is not regulated well, and too much acetylcholine is released instead of regulating it, it can also be associated with forms of schizophrenia too. And that is a condition in which an individual sees and hears things that just aren't there. And they do know that there's an overproduction of this neurotransmitter, acetylcholine, and it's not well regulated by the dopamine. That can happen. But those are the basal nuclei. And I do have a transparency of that one: stroke, cerebral vascular accident, transient ischemic attacks, and Parkinson's disease. Concussion is a jolt to the head in which an individual can be knocked out temporarily and eventually come to. They do know that people also that have had concussions, especially men who've had concussions, can develop or have a higher incidence of amyotrophic lateral sclerosis, and that is the condition that Stephen Hawking had. And what it does is it just destroys a lot of the motor activity gradually to the rest of the body, and their mind stays perfectly intact. They just lose all total, complete control over the muscles of the body, and blood is poured out into the surrounding nervous tissue. Nervous tissue is very, very delicate, and if it comes direct contact with blood, there are some hormones and amino acids in blood that can actually damage the nerve tissue. So that would be a broken blood vessel. Many times you can look at the outside of a fresh brain, someone who has suffered a stroke or many strokes, and you can see where the blood has pooled on the outside of the brain and done damage to that neural tissue. Another thing that can happen is the blood vessel doesn't have to break, but it can bubble out. The wall of that blood vessel can weaken and it'll bubble out. When it bubbles out, attacks. These are tiny strokes, very, very tiny capillaries may break, and that individual may have difficulty orientating or thinking or verbalizing for a few moments, and then they come back to their senses. And this can happen a number of times. Sometimes that's a warning, a transient ischemic attack. It's a warning of nuclei and the relationship between their secretion of dopamine, which controls adequate amounts of acetylcholine for those smooth movements. Okay, now let's look at lecture 5. And in lecture 5, we get to talk about ventricles and the cerebral spinal fluid. Now, let me see. Okay, this is on page, the word ventricle means a little space or a little room. And the next, you hear about ventricles in the brain, and you also hear about ventricles when we're talking about the cardiovascular system, because the two lower chambers of the heart that receive blood from the upper two atria, when it pumps down into the ventricles and then goes out the arteries. The lower chambers of the heart are also called ventricles. But these ventricles are small chambers in the brain. There's 4 of them in number. There is one in either hemisphere, one in the right in the middle of the diencephalon, one behind the pons and in front of the cerebrum. These are 4 rooms in the brain filled with cerebral spinal fluid, CSF. This cerebral spinal fluid is very important. And I think I mentioned to you earlier, it works to cushion the brain, just like the fetus is cushioned in the uterus, and any kind of bumping can protect that fetus. All of this cerebral spinal fluid cushions the brain. The brain floats in the cerebral spinal fluid. If you take a fresh brain out and just lay it down or even hold it in place, the neural tissue is so delicate that it will become flattened wherever you're holding it. It'll flatten on the base portion of the brain if you're sitting it down in a dissection pan. So this cerebral spinal fluid enables it to float within the cranial vault. Now, the cerebral spinal fluid comes from the plasma of the blood, but it's different than the plasma of the blood. If you look right here, this is an exaggerated picture of the ventricles of the brain. This would be the left ventricle, and here is the left ventricle. This would be the left horseshoe-shaped ventricle here in the left hemisphere, and it's referred to simply as a lateral ventricle. You have one in the left hemisphere, you have a lateral ventricle in the right hemisphere. It's a C-shaped ventricle, and they've swollen it quite a bit here so you could see it. That is the lateral ventricle. You have 2, and the cerebral spinal fluid from the lateral ventricles flow into the 3rd ventricle, and then from the 3rd ventricle down into the 4th ventricle. The fluid not only exists within these ventricles of the brain, but as it flows down into the 4th ventricle, it will flow down through the middle of the spinal cord and in what we call the central canal. It'll also flow all the way around the entire brain, and it also flows all the way down and around and up through the spinal cord. That's why we call it cerebro. It floats in this cerebral spinal fluid, cerebro and spinal fluid. You're going to be surprised when I tell you you only have maybe 5 or 6 ounces altogether, not even a complete cup of cerebral spinal fluid. And yet it is enough to keep the brain protected and buoyant, as well as the spinal cord, so that neurons don't settle one upon the other and that they're not damaged because they're buoyant within this fluid. The cerebral spinal fluid is very special. It does come from the plasma, and neuroglia that we have mentioned before, there's a special kind of neuroglia called the ependymal brain. We call it the choroid plexus, and I'll mention this quite a bit, but these capillaries exist in the left and right lateral ventricle. The capillaries exist in the 3rd ventricle and in the 4th. And these capillaries, we call the cluster of capillaries the choroid plexus. In that choroid plexus the present in that is the ependymal cells. And the ependymal cells are the cells that actually filter from the plasma the contents of the cerebral spinal fluid. Now, the cerebral spinal fluid floats the brain, protects the brain, cushions the brain, but it's also nutritive. It does have lots of nutrition that your neurons need. The neurons of your brain obtain their nutrition from the fluid, cerebral spinal fluid, yes, but most of the nutrition comes from the brain capillaries, the carotids on the right and left side of the neck. The carotids come all the way up the neck. They bifurcate. You have an external and an internal carotid, and these carotids break that the exchange is made and the neurons are fed. But the cerebral spinal fluid, as I said, is also nutritive. Now, you actually make about half a liter or more a day, and you drain half a liter. That means you're constantly making it, and your brain and within and on the outside and within the cord, on the outside of the cord, they only have about 5 to 6 ounces. But this cerebral spinal fluid is draining back into the blood, and then new cerebral spinal fluid is filtering in. It is the choroid plexus, the capillaries that are in the lateral ventricles and in the third and fourth that extract from the plasma the contents of the cerebral spinal fluid. It's very important because there are certain hormones and amino acids that are naturally in the plasma of your blood, and they do fine with your body tissues, but they can't come into contact with the brain. So the ependymal cells, those little neuroglia, extract from the plasma this fluid and sends it through these little blood capillaries known as the choroid plexus. And there's pictures of it in your textbook and a good description. I will give you those page numbers when I email you when I'm through. Now, if you look right here, I'm going to hold this up. I wish this was more dramatic because I would love to show it on the overhead, but you can show you how it is formed and how it is removed. So this would be to be on the other hemisphere. And inside here, the little capillaries are extracting from your plasma the CSF. This cerebral spinal fluid fills up this lateral ventricle, and some of it drains from the base of the lateral ventricle through a little canal right here, or a hole with that permits the fluid to flow through that canal, and that is referred to as the interventricular to this egg-shaped hollow structure known as the thalamus. The inside of the thalamus is hollow and it contains the cerebral spinal fluid, and there's even a choroid plexus here which has capillaries that are making the CSF from the plasma. The cerebral spinal fluid then flows from the third ventricle, this is the first and second, down the third through the interventricular foramen, into the third ventricle, down the fourth to the fourth ventricle here, that is in front of the cerebellum, behind the brainstem. This is the fourth ventricle. You can see that on that page. And this long canal is referred to as the cerebral aqueduct. All of these are named from the Romans. And the aqueducts, if you remember from studying a little bit about Roman history, the aqueducts were canals around the Roman cities in which the water was pumped, and then it was pumped down into wells so that the citizens could come and gather the water on a daily basis. So this is the cerebral aqueduct, which takes it down the fourth ventricle. In that fourth ventricle, there's a hole in the floor of the fourth ventricle, and the hole all the way around the entire brain. It'll flow down in front of the spinal cord, behind the spinal cord, and then through the central canal of the spinal cord. Now, you may wonder, well, Miss Angel, you've just told us that the choroid plexus these blood vessels in the ventricles, they they bring in the cerebral spinal fluid with the help of the ependymal cells, but it drains because you never have any more than 5, 6 ounces of cerebral spinal at any one time. You're constantly making it, constantly draining it, kind of like a jacuzzi, where they from the plasma and drained back in. And there is a structure we'll talk about later called the arachnoid villus, little finger-like structures that reabsorb that cerebral spinal fluid in the same amounts that it's made. If something happens and the interventricular foramen or the cerebral aqueduct is blocked and the fluid does not adequately drain, if it builds up within these ventricles, that's referred to as hydrocephaly, water on the brain. When there were times when babies were born, if they developed hydrocephaly, are movable. Those are our synarthrotic plates of the skull, which are movable at birth. And by the end of the first year of life, they become immovable. If a baby develops water on the brain, that brain will get larger and larger and larger, that skull, because those two Arlington Developmental Center, and I saw a little boy that had hydrocephaly, and the head was quite large, where the body itself reabsorbs it. So they definitely do have a treatment. But if that doesn't drain adequately from form. Okay. Let's look at the brain stem. Okay, I'll use that one. Oh, before I do that, before I get into that, I want to show you another transparency that if I had your beautiful faces in front of me in class, I could show it on the overhead because it explains it too. Now, this is the choroid plexus, and this is in Hole's Human Anatomy and Physiology. So you can find it here, and all you have to do is look for the choroid plexuses in the ventricles. And look at that. This was figure 11.4. I believe this was on 373. But here is the third ventricle. Right up in here would be the left and right lateral ventricle. The fluid flows through the interventricular foramen here into the third ventricle, from the third down into the fourth, and you see the arrows going all the way down, and then they show you the bottom, all the way around the entire spinal cord and back up. And that's showing you also how this flows. It also shows in here a little area where the blood vessels are kind of clustered. And that cluster of blood vessels containing also the ependymal cells, those are what we call the choroid plexuses. And this would be the choroid plexuses of the ventricles. You see blood vessels which extract the CSF from the plasma itself. And that's a good diagram. Now, the brain stem. Remember, there's an argument. When you have two anatomists, you have 5 arguments. Some consider the brain stem to be the diencephalon, as well as the midbrain pons and medulla. And this was also in Hole, a transparency. So here's the cerebrum, the cerebellum, and they included the diencephalon with the brainstem. So, and they included here the brainstem is the diencephalon, the midbrain pons, and medulla oblongata. So some separate it and some don't. Now I want to orient you a little bit. Here is the the diencephalon, here's the thalamus, an egg-shaped structure below the corpus callosum. It's gray in area, in gray in color, I'm sorry. It's gray in color because you're seeing thousands and thousands of naked dendrites and cell bodies. This is hollow, and it is filled with cerebral spinal fluid. The inside of the thalamus is the third ventricle. The walls of the thalamus are the neurons that carry on the activity, and I'll give you the physiology soon. Hanging from the roof of the thalamus is a little gland right here called the pineal, Pineal, because it looks something like a pineapple. And this artist has illustrated hanging off the back of the thalamus, the pineal gland, and sometimes you'll see an artist illustrate them hanging from the roof of the third ventricle. The pineal gland, the word pineal comes from pine because it's kind of pine cone. And the pineal gland, don't confuse it with the pituitary right here. Please don't confuse that. The pineal gland, hanging from the third ventricle, secretes a hormone known as melatonin. And melatonin is what our body builds up when the sun goes down. The pineal gland releases the melatonin, and the melatonin is what gives us our sleepiness, and we can go to sleep. That's why they say many times you can sleep much better in a dark room, because in that dark room you'll release more melatonin and it'll put you to sleep. The Nazis in World War II experimented with the pineal gland on young children and babies. It was terrible, but they did. If this room was the thalamus, the ceiling would be the epithalamus, the walls would be considered the thalamus, and the floor, the hypothalamus. And off the hypothalamus, you have a stalk of tissue called the infundibulum, and from the infundibulum, the pituitary gland. So the diencephalon is many times considered the thalamus, the hypothalamus, the infundibulum, the pituitary, and the pineal. Below the thalamus is the midbrain, and you can see it. It kind of looks like the lateral view of a duck's head. There's the beak and there's the head. That is the midbrain. Below the midbrain, this tissue that bubbles out and is right in front of the cerebellum is known as the pons. And below the pons is a bubbling out of tissue, also known as the medulla oblongata. As the spinal cord ascends through the intervertebral foramen and enters the foramen magnum, which is that large hole we studied at the base of the skull, that is where the spinal cord enters the brain. And right at that spinal cord, as it ascends, it bubbles out a little bit, forming the medulla oblongata. And it's on the front portion of the medulla oblongata that the tracks, we call them tracks because it's in the central nervous system of the brain. I think I may have said nerves cross over, but the tracks cross over on the anterior portion of the medulla oblongata. And those tracks, we say they decussate. And the right brain handles the left side of the body, and the right side of the body, right there at the decussation. On page 16, there's a discussion, a little bit more. I'm going to fill you in on all of this. The diencephalon, we'll talk about that. We'll talk about the thalamus and the hypothalamus. Now, the diencephalon, I have already mentioned the thalamus, hypothalamus, infundibulum, pituitary, pineal gland, all of this. And they also include the optic tracts in that, and the optic chiasma. We'll learn about that later. But all of that's considered part of the diencephalon. And the diencephalon, by some anatomists, is referred to as the interbrain or the tween brain, because you cut down through the corpus callosum when you separate the two hemispheres of the brain, And when you cut down through that corpus callosum, there is the diencephalon, and that's the structure they're referring to as the interbrain or the tween brain. Now, let's talk about two structures specifically, the thalamus and the hypothalamus today. The thalamus, remember, is that egg-shaped structure below the corpus callosum, hollow on the inside, containing the third ventricle filled with cerebral spinal fluid. But the epithalamus, the ceiling, and the walls of the thalamus are important. As I said before, it's gray in color because you're looking at naked dendrites or non-myelinated dendrites and non-myelinated cell bodies. The thalamus, if we talk about its physiology, now you know where it is and you know its structure. We talk about its physiology. That thalamus is kind of the policeman that stands in the middle of the road. What is the biggest intersection in town? I guess Highland and Poplar. Okay, when those lights are out, they send a policeman out and he has to direct the traffic. And he permits east-west traffic to move. He stands in the middle, blows the whistle, and tells them to come. And then after a few cars have passed by, he stops them. And then he lets the Highland traffic come across going, is a traffic cop. He directs the flow of traffic. Well, that's what that egg-shaped thalamus does as well. It directs impulses. It directs all impulses coming from the brain that are sent down through the diencephalon to the brain stem, all the way down the spinal cord and out through nerves to muscles and glands. That thalamus sends it to the right area. Anything that comes up from the brain, from the spinal cord through the brain stem, for example, tactile sensations, how your clothes feel on your body, your knowledge of where your muscles are in space and time, it's going to come up through that spinal cord, through the thalamus, and the thalamus routes, because that's it. That's a good word for it, router. It routes that message. It routes that track and sends the message. If it's the way your clothes feel, it's going to send it to the post-central gyrus right behind the central sulcus. Everything that you see, your vision, will go through the thalamus and be directed to the occipital lobe of the brain. Sound will be routed. Those sound waves will be routed through the thalamus and directed to the temporal lobe of the brain where sound is perceived. So the thalamus routes everything and sends it to the proper area. So everything you're seeing, hearing, tasting, touching, all of that is routed through that thalamus to the right part of the brain. And that gives you cognition of what's going on in your environment. And then you manipulate the environment from the motor activities coming down from the precentral gyrus down through, of course, the basal nuclei. They release dopamine, regulates acetylcholine, which comes down through the thalamus. And then it's going to go down the rest of the brain stem and out to the arms and legs so that you have volition over those muscles that control your arms and legs. So think of that thalamus as a traffic cop. Now, there's only one sense that is not routed through the thalamus, and that one sense is olfaction, your sense of smell. Smell does not go through the thalamus. Everything else does, but not smell. Your sense of smell comes to you because of your sensory afferent neurons that pick up chemicals that dissolve in the nasal mucosa, in the mucus of your sensory neurons in that membrane, and it takes it by large olfactory tracts deep within the brain, so you have that awareness of odor, that sense of smell. It bypasses the thalamus. Everything else goes through the thalamus. it bypasses that thalamus. Be sure you remember that. Word to the wise. Now, there's some interesting things. The greatest, and a few years before he died, he would sit with a group of friends and talk, and he said, them could smell it. And he said at times it smelled like garbage, but there was none around. Well, later on, it was discovered that he had a temporal lobe tumor that impinged upon those neurons for the sense of smell. And the tumor was benign, I believe, but it had infiltrated the brain. It was way too far gone to save him. It would have been wonderful if we could have, because he was such a wonderful composer. But diagnosed it. We do think that the sense of smell was probably one of the first senses that animals were able to obtain from the environment. We evolved that first sense of smell, and sense of smell gives us many other animals a reason to develop the sympathetic response. A deer can smell a cat. They can smell a mountain lion that may have been in an area several days before, and they're alerted to the smell of the ammonia where the cat had urinated, and the deer can pick up on that and flee. And many times they can smell an enemy before they can actually see or hear, even though their hearing is very good. So we think that the sense of smell, olfaction, was probably the very first sense that was ever developed. After the thalamus, let's look at the floor of the thalamus, which is the hypothalamus, floor. The hypothalamus is interesting because the neurologists and the endocrinologists argue back and forth over it. The neurologists say it is truly an organ that needs to be incorporated in the nervous system. And because it is an organ that controls the autonomic nervous system, all those things that we don't have to think about are controlled by the hypothalamus. In your notes, heart rate, blood pressure, body temperature, water, electrolyte balances, hunger, body weight, sleep, wakefulness, all of that is due to the floor of the thalamus, the hypothalamus. And so because it's the brain of the autonomic nervous system, the neurologists claim it. Just like our conscious brain is the cerebrum. And with that cerebrum, we control our environment and what we do. Well, that's the conscious brain. The unconscious brain can be directed by the hypothalamus and has all those autonomic activity hormones, which are also released hormones. And these are protein hormones. And most of the hormones in your body are protein hormones. The steroid hormones are the testosterone, estrogen, progesterone, cortisone. Those are steroid-based hormones. But most hormones in your body are primarily protein hormones. And the hypothalamus releases 10 hormones, protein hormones, that we discuss in endocrinology, important ones. So the hypothalamus is a mass of nervous tissue with these endocrine glands embedded. And the hypothalamus is full of blood vessels. So these endocrine glands or cells in the hypothalamus will release the hormones directly into the bloodstream. Now, is released, a mother during the later stages of gestation will start to release oxytocin, which gives her the mothering hormone, so that she will bond to tuned to their wives during that pregnancy. They can produce that oxytocin too. Now, oxytocin is also produced in great amounts after the fetus is born, and it's what they call the milk letdown factor, because it's the oxytocin that causes the mammary glands it will do that. So oxytocin, feel-good hormone, it's made by that hypothalamus. Mom bonds to the baby and dad does too, the colostrum, and then the milk comes down. The other one is ADH. I didn't write that out. That's anti-diuretic hormone. Anti means against. Diuretic. A diuretic is something that makes you urinate. Coffee and tea and Cokes are diuretics. Well, the ADH, antidiuretic hormone, which is made by the hypothalamus, is released into the bloodstream and it goes to your kidneys so that your kidneys release the appropriate amount of urine a day, which can be anywhere from 1 liter to 1.5 liters. Now that sounds like we lose 1 liter to 1.5 liters, that we need to drink 8 to 10 glasses of water on a daily basis. But ADH enables you to release enough water to remove the nitrogenous wastes from your plasma, release that fluid as urine, and then retain the excess fluid so that you don't become dehydrated. So ADH is released when the temperature is very, very warm. You're going to naturally perspire. You're losing fluid through the skin, and your body doesn't want to release too much fluid through the urine, so your hypothalamus makes that ADH, so you retain it. If ADH is not released appropriately, and they call that disease insipidus, and you're not making enough ADH, and if you don't make enough ADH, a patient can actually urinate to death. They'll dehydrate. They cannot retain fluid. It's easy to remedy. You just give the patient ADH, a little tablet of ADH on a daily basis, and that suffices to make sure that the patient doesn't urinate too much, that he retains the needed fluid in his body and releases through urine the fluid that needs to be released. So that's oxytocin and ADH. Those are only 2 of the 10 protein hormones that the hypothalamus releases. So we talk about the hypothalamus in conjunction with neurology and endocrinology. And that's one of the first systems that you'll study in AP2, a fascinating system. So what did we do today? Well, we finished lecture 4 and 5. We talked about the physiology of the cerebral cortex. We discussed the motor activities from the precentral gyrus in front of the central sulcus. And we talked about the tactile sensations in the postcentral gyrus. We also mentioned memory and reasoning, analysis and personality from the frontal portion of the frontal lobe. And we even mentioned the Broca's area usually in fact, I'm confused, in the left hemisphere. Sometimes it'll appear in the right, but Broca's area is a concentration of neurons that give us our ability to enunciate words in proper speech. Okay. We also mentioned that you think with your entire brain, and it's the association neurons, also known as the interneurons or connective neurons, that link the sensory to the motor and make sense of what we're experiencing. And they're scattered throughout the entire brain. We discussed again the corpus callosum. You cut down through that corpus callosum, and what do you enter? You enter the diencephalon, which was the thalamus, the hypothalamus, the infundibulum, which is a stalk of tissue, at the bottom of which is the pituitary. This is the diencephalon. And we talked about the basal ganglia to. And the basal ganglia are deep, they're darker regions deep within the brain that release that important dopamine, which regulates acetylcholine. A lack of that dopamine results in Parkinson's disease. And we found out that an excess of dopamine, possibly an excess of acetylcholine, can result in schizophrenia. We mentioned a little bit cerebral injuries, and that was also on page 14. We discussed in lecture 5 the ventricles, 4 ventricles, and the ventricles contain cerebellum, the outside of the spinal cord, the 4 ventricles of the brain, and goes all the way down the central canal. It makes the brain buoyant, and it also feeds the brain. We know that the cerebral spinal fluid is extracted from the plasma by specific ependymal cells in capillaries, known as the choroid plexus, inside of these ventricles, and that choroid plexus removes certain hormones and amino acids from the plasma, and that is absent in the cerebral spinal fluid because those hormones and certain amino acids can cause damage to the brain. We know that the choroid plexus, with that ependymal cells, they make that cerebral spinal fluid, and the arachnoid villus drains it. Make sure you can trace it. We talked about the right and left lateral ventricles drain to the 3rd ventricle, which is inside the thalamus, by the interventricular foramen, and the 3rd ventricle drains down a canal known as the cerebral aqueduct into the 4th ventricle. There's a hole in that 4th ventricle through which the fluid flows around the posterior part of the brain, the anterior part of the brain, and flows down the central canal and around the outside of the spinal cord as well. We talked about the brain stem. We mentioned and located the diencephalon, midbrain pons, and medulla. We'll talk more about the responsibilities of those structures in the brain stem a little later. We did talk about the thalamus and its responsibility. It's an egg-shaped gray structure found below the corpus callosum. It's hollow because it contains the 3rd ventricle filled with the cerebral spinal fluid, the brain to the proper location. So everything you see, hear, taste, touch, feel, even proprioception goes to the proper portion of the brain because of that thalamus. And all except one, and that is the sense of smell, olfaction. Olfaction. That's a straight shot to the temporal lobes of the brain, deep within the brain, so you have your sense of smell. Straight shot. It bypasses that thalamus. Then we talked about, everything coming from the brain too. All your decisions in the precentral gyrus in front of the central sulcus as to where your body's going to be in space and time, moving it, that's going to come down through the thalamus, which will route it down through the midbrain pons, medulla, down system. We mentioned the hypothalamus, which is the floor of the thalamus, and we said it is the brain of the autonomics. All those things you don't have to think about. They're in part controlled by the hypothalamus. You get into endocrinology and AP2. We just mentioned 2 of them here, and that was ADH and oxytocin. So, goodness, 6:30, 7:30. I've been going an hour and 15 minutes. So it's a lot of material. I'm going to email you, so please study these 2 lectures, 4 and 5. Listen to the presentation. Read the pages that I'm going to direct you to when I send your email. And also let me know. Let me know. You can call me and I'll check you off, or you can email me that you have done everything and you are up to par through the 5th lecture. You've listened to the videos and you've studied this. You've read your textbook and you've answered the pertinent questions in the study guide. So please let me know. I can check you off. And when I check, so that will be 100 points right there. And I'll depend on your honesty to let me know. This is all material that you're using it all the way through your subsequent classes. So please, please put in the time. It's interesting material. It's fun to teach. It would be more fun if I had your bodies in front of me. I do miss you guys. I don't like this at all. I like being in class with you. So let me know by email or call me that you're current, you're up to date on all of your work. Thank you for spending the time with me. And until next time, I will end stream. Bye-bye.